In a world where plant diseases threaten our food security and ecosystem stability, the importance of accurate, efficient, and rapid plant disease detection cannot be overstated. This video shows how a small improvement in artificial neural networks can help with plant disease detection. For a while now, deep convolutional neural networks, or DCNNs, have been successfully utilized for plant disease detection. The classic approach involved feeding a DCNN with one branch red, green, blue, or RGB color coordinates. However, there is a better and more efficient approach. Instead of RGB color coordinates, a new proposition sees the DCNN being fed with CIE Lab color coordinates. Picture this as a modified version of the Inception V3 architecture. It includes one branch for achromatic data, which is the L channel, and another branch for chromatic data, which are the AB channels. This ingenious modification capitalizes on the decoupling of chromatic and achromatic information. What does this mean for the future of plant disease detection? Well, for starters, splitting branches reduces the number of trainable parameters and computation load by up to half of the original figures using modified layers. It's a step up in efficiency, managing to achieve a state-of-the-art classification accuracy of 99.48% on the Plant Village dataset and 76.91% on the Cropped-PlantDoc dataset. This new architecture divides the first three layers of the Inception V3 model into two branches, one for the L channel and another for the AB channels, computed from the input RGB image. Then, the output from each branch is concatenated to follow the rest of the network as in the baseline. In essence, the two-branch DCNN proposed in this study specializes in learning chromatic and achromatic features from the CIE Lab color space. This results in a more efficient and effective plant disease classification. The experiments conducted prove that this approach outperforms the classic one-branch RGB images fed DCNN. It does this by saving a portion of learnable parameters and floating point operations, reducing the numbers from 1/3 to 1/2 in those initial three layers. This is achievable because the RGB channels are highly correlated. Hence, working in a decorrelated color space avoids redundant filter weights. In conclusion, this new approach proposes an optimization along the first three convolutional layers of usual DCNNs, where the first three convolutional layers specialize in learning chromatic and achromatic features from the CIE Lab color space, while saving a significant portion of learnable parameters and floating point computations.